Okay, well, thank you very much, Heinz and Thermo, for inviting me. I'm going to tell you about the identification of unknown compounds using high-resolution mass spectrometry. What I want to do is start off with acknowledging the co-authors. I'm up here talking to you today, but most of the work has been done with a number of other co-authors, focusing mainly on the work from Li Shen for the identification of flame retardants and some of the high-resolution mass defect plot work by Carl. Presented this paper in 2006, showing there's almost 100,000 chemicals of commerce, and a number of them are produced at very large amounts, a million pounds per year. But we only analyze a very small amount. This is the Stockholm Agreement list. It's got 12 compounds that were the original 12, a series of others added in 2009. And there's a few that are currently under discussion. And so in terms of regulations, there's very few that are monitored. One of them, as I said, the original 12 included a lot of pesticides. And if you can see this series of pesticides here, they were made by a Diels-Alder addition of hexachlorocyclopentadiene to another double bond, and the majority of these compounds were manufactured that way. In the early 50s, these pesticides were considered to be good. This is from Washington. It was an ad that crop yields were increased by using pesticides, and it was a good thing. This is another ad. It's from the Smithsonian in Washington, and it says at that time a $25,000 reward for the first bug that doesn't or isn't killed using real kill, which is dieldrin. In today's dollars, that's like $250,000. So these things were very toxic and very persistent in the environment. And Rachel Carson wrote Silent Spring, and one of the reasons it was Silent Spring is because birds were eating worms that were moving through the decaying foliage, and if this continued, we would not have any birds anymore. So in the early days, and you know, Dawn, did you show? No, it was yeah. And so this is one of the early chromatograms. This is from a paper by Sorensen, and there was these unknown compounds, and identifying unknowns have been a real challenge. 13 or 14 of these peaks were not identified, and this is for the analysis of DDT. It took a long time to discover that these original compounds were actually PCBs, and Jensen had to analyze eagle feathers back to 1888, and he started to see these compounds in the 30s. PCBs were used in a whole bunch of applications, heat transfer fluids, additives, starting in the 30s, and it wasn't until they were using GCMS in the early 60s, and from roughly about 1958 is when the electron capture detector was developed, till the mid-60s. It actually took about 7 years to identify these compounds as PCBs. So identifying unknowns is a challenge. And this is the paper by Sorensen. It's in Ambio, and it's a very interesting paper if you get a chance to read it. 1972, so the reference is down the bottom there. As I said earlier, identifying unknowns is quite a challenge. We typically have to separate them from the matrix. A lot of times there's very many isomers. You need to have good chromatography, and I'm going to show you examples of multidimensional chromatography that help separate out these compounds. You need to get a clean mass spectrum in order to try to identify them. The use of high-resolution mass spectrometry and mass spectrometry is typically used, again, to help deduce structures from first principles. It requires extensive detective work, and you're looking at things like patents, documents from manufacturers. The InChem website is a very good website that has background information, and once you're able to do that, then hopefully you'll be able to synthesize the compound for confirmation. So typically when we're looking for unknowns, we have target compounds, so that would be taking many of the compounds that we look for. We can look for targeted unknowns, and that's taking some of the compounds on the Muir-Howard list or in patents or other products. And so those are fairly easy to try to identify. And then there's untargeted unknowns, compounds that we don't know, we come across, and that happens to us quite often. And they can be from a number of different sources, including degradation products or reactions with other compounds. So just to give you an idea of how complex that could be, this is a sediment from Lake Ontario, and you can see that it has a significant number of peaks in it, and trying to identify compounds are a real challenge. Typically what you have to do is separate these compounds because they can have many different components. Dioxins have 210, PCBs 209, compounds like toxivine have hundreds. When you're looking for it, you want an idea of the toxicity, and so that's a subgroup. That's why you have to separate it. In many cases, the toxicity can range by orders of magnitude. Concentrations can go from, as Dawn showed us earlier today, the femtogram levels, potentially up to percent levels. And the different matrices have all very different interesting idiosyncrasies that you typically have to deal with. And what you want to do is take these samples, clean it up so you can get it in a GC vial in order to analyze it. And sometimes, as we've heard earlier today, that can be a big challenge to make sure you've got a clean extract that you can quantify. So just to give you an idea, this is the sediment sample that I showed just a couple of slides ago. And if you want to look at something like PAH, you type in the mass and you can see it. And in this mess, actually, these area counts are about a couple of hundred compared to almost 10 million. So they're just a very small fraction of what really is in this sample. Same with other targets like PCBs or some of the pesticides. We're very good at cleaning up matrices and then identifying and quantifying compounds we know. But what we're not good at is separating and identifying unknown compounds. So most of us have just come from the flame retardant meeting, and there's a lot of different brominated flame retardants. Our group has done a lot of work, especially Li Shen, on the chlorinated flame retardants, and I'm going to spend the next little bit talking about how they were discovered and some of the work that we're doing on the degradation products from these compounds. We originally started looking at the chlorinated flame retardants because of Dechlorane Plus. It was originally discovered in Great Lakes Sediment by Ho, who was a postdoc with Ron Heights. Dechlorane Plus is still manufactured by OxyChem in Niagara Falls, and it's used in a lot of cabling, wires, and connectors and in carpeting, and it is a high production volume chemical, so it's made at more than 1 million pounds per year. So when you look at some of these retro Diels-Alder or Diels-Alder pesticides, the main peak in the mass spectrum is a retro Diels-Alder, which is the separation of the hexachlorocyclopentadiene at 272, and that's typically ions that we monitor. So Myrex does that. So does SYN and anti-DP. But when we looked at it, we had a series of other peaks, and so obviously they were this type of compound, but they were unknowns. And as I said earlier, you go to the literature and you try to find out some background information. And this is a document from the World Health Organization. And you can see that it says Dechlorane Plus was used to replace Mirex, and so were some other compounds like 602, 3, and 4. So we went and tried to get an idea what the structures were, and these are the structures of Dechlorane 602, 3, and 4. We then looked around a little bit more and to see where they would be used. 602 is an additive in fiberglass or nylon, used in high voltage cabling, and it's typically used in combination with 604. And 604 is used in Molykote, a silicone grease, for temperature stability. We couldn't find any application of 603. So Lee looked on the chemical registry, and it tells us that 602 is coming into Canada in some domestic products, so is 604, and Dechlorane Plus is a high production volume chemical at more than 1 million pounds per year. So in order to confirm that some of these peaks may be these flame retardants, we had them synthesized. But I guess the background is that we saw them at higher levels coming out of the Niagara River. There was a source there, and we were able to have clean peaks when we were looking at the 272 peak. And so we synthesized them, or had them synthesized, and then what we did is we wanted to try to match the retention times. So this was the standard, 604, 3, and 2, and this is the sediment from Lake Ontario, and you can see that the retention times match very well, which confirmed that that's what these compounds were. We then took a look at sediment cores just to get an idea of the historical use of these compounds. And so Mirex appeared in Lake Ontario sediment around 1950. So did 603, and another compound that was similar, Chlorine Plus, and I'll show that to you in a minute, while 602, 4, and Dechlorane Plus started to appear later. There's a little bit of a blip in here because around 1985, Hooker Chemical put in a process to try to recover a lot of the fugitive emissions, so that's kind of consistent with the levels getting lower, but they're making more of these compounds now, especially that some of the brominated flame retardants, BDEs, are restricted. But you can see that 602 was started to appear earlier, and so did Chlorine Plus. And when we took a look a little further, as I said earlier, we were able to identify where 602 and 4 came from. Lee obtained some dieldrin and aldrin and found it as an impurity in those pesticides. And she also found that Chlordine Plus, which is this compound here, was present as an impurity in chlordane and chlordine. So we continued to look for these compounds, and some of the work that Lee has done, you can see that we're seeing peaks at 272 and 237, and what that is the chlorine is exchanged with a hydrogen. This is like photomyrex. There's a photochemical reaction where that happens, and then what you see is you have these two fragments, and when you run a sample, you can see there's two of these in basically the two isomers. There could be an endo or exochlorine, so one or the other, and that's why you have two. So Lee checked to see if the molecular ion matched, and they did, and we had some industrial or reaction industrial standard made, and so Lee took a look at some of these in fish, so he was able to see the two compounds in fish. She also saw some other compounds, which was actually the loss of two chlorines, and but these monohydro Dechloranes, as the loss of chlorine and replaced by hydrogen, were present in the sediment lower but much higher in fish. So these compounds appear to be more bioaccumulative than the parent product. We went and did a number of analyses of fish samples, and you can see these are from Lake Ontario. As you move away from the source, and the source is right here in the Niagara River, the levels go down, but the ratios of the photodegradation products, which is the yellow and the green, go up versus the parent product. And you can see that happens as you go away from Lake Ontario, the source. So consistent with these compounds being photodegradation products. And the ratio of the alpha beta, that's the first and the second peak, it changes slightly. One is more stable than the other. So we wanted to see how, if we're detecting these in the Arctic, we obtained samples from Greg Tomey and Rob Letcher, beluga and polar bear samples. The samples were analyzed, and Dechlorane 602 Myrex and photomyrex was detected in all of them, but not DP or 604. And Lee was able to identify the beta 602 degradation product, but not the alpha. So it appears that the beta is more stable or being formed at a greater rate. So as I said earlier, we had a technical mixture that was produced. So these are the monohydro, and then these are the blue ones are the dihydro. Lee was able to detect only one of the monohydros in the Arctic beluga and two of the dihydros, and then in the polar bear is the same thing, but she detected different dihydroisomers. You can see here in the polar bear the degradation product and the 602 are roughly at the same concentration, but as you move away, the ratio goes up, indicating that this is likely a photochemical product. And one of the interesting things is 602 is fairly bioaccumulative. Levels of 602 versus Myrex were roughly the same in the polar bear that Lee analyzed. So I'm going to change direction a little bit. I'm going to talk about compounds that were produced in a car parts recycling plant fire. So this was a fire that burned in 1997 in Hamilton, Ontario. There were about 400 tons of car parts that this plant recycled, dashboards, car seats. So it had a whole bunch of flame retardants in it, and you can see the firefighters. It burned for about 4 days. As I said earlier, several of the firefighters have had serious health problems. The fire was so bad it produced a lot of hydrogen chloride, so that's one of the first things that came up, that the bumpers on the fire trucks just corroded off. So it was kind of a nasty. You can see the photos from around the neighborhood. So we extracted one of the ashes, and again, like the sediment I showed you before, this was an extremely challenging sample. And so what would you do if you get a sample like this, and you kind of think of where would you even start? And our lab has been doing a lot of two-dimensional chromatography in the last few years. Don mentioned earlier, or showed a schematic. And so it's basically two columns at two different phases connected by a modulator. And what it does is it traps and releases, and because you're putting it in a second phase, you get a different mode of separation. And what you get is little slices. You can plot this and in the form of a two-dimensional chromatogram. Typically what you want to do is set up the system so that it's orthogonal, and that is the first column and the second column separate your compounds in completely different ways. Normally the first column is a longer column, and the second one is much shorter. It can be a thinner phase or not, and that just depends on what separations you want to do. So this peak modulation, typically you take a peak as Don showed earlier. It's chopped into little slices, and the slices are narrower and taller. So the way you get extra separation as well is that you've got a lot more space to put other peaks in, and your peak capacity can increase by an order of magnitude or more. So this kind of just shows peaks that were co-eluting. You break them into little slices. You actually turn them 90 degrees and you plot them either in a contour plot or a color intensity plot. And I'll show you some examples in a second. This is the hardware. You have the primary column goes in through, this is the modulator. It's just two hot and cold jets that trap and release into the second column. And in this instance, the mass spectrometer is over on this side. And so what you're able to get is two-dimensional spectra. This shows some challenging PCB separations, 81 and 87, 149 and 118, or 110 and 77. So you're able to separate them. You can see the little white in the back here, which is 4. It looks as 4 peaks, but it's really 8. So you get significantly enhanced peak capacity, especially with many compound groups that have lots of isomers like PCBs. And if you set it up right, this is an RTX PCB in the second dimension and an RTX1. You can pull out all the dioxin-like PCBs. You can see that they're eluting later, and at the top, well, all the rest of them are down here. Organochlorine pesticides, again, you're able to separate these compounds, and a lot of labs do dual column work, but you're able to do this in one injection and separate them out. And one of the really interesting things about PCBs is that, again, this is RTX PCB and RTX1. They can align in these bands, and when they align in the bands, these are all the same degree of chlorination. So if you look at it in 1 degree separation, you can have a lot of chromatographic traffic. This just kind of fans things up, separates them out, and now you've separated many more peaks. And we have a method that we look at with electron capture detection as a screening technique, and it allows us to do a lot of compounds at once. We're able to do chlorobenzenes, PCBs, and organochlorine pesticides, chlorinated paraffins. You can see they separate out in bands, mostly by degree of chlorination, and chlorinated paraffins is one of the more challenging analysis. PCNs and chlorinated diphenyl ethers, dioxins, and this is listed as an unknown, and that's just because we didn't have a standard. When we put on a mass spec, it was actually chlorinated terphenyls. So you're able to identify this and use it as an analytical triage and get an idea of what may be in your sample. So this is that sample, plasmid sample that I showed you a while back. And even in a 2D plot, it can be really complex. So in this plasmid fire, there was over 850 aromatics and many aliphatics. Most of the aromatics came across this line and the aliphatics, so you can set it up to separate. And taking a little slice here, you can see there's a whole bunch of other different compounds, a number of different PAH that Dave talked about earlier. And this is PCNs. So again, the PCNs, the homologous series, tend to separate out in bands, which makes it much easier to identify and quantify. But one of the problems is, even with this enhanced chromatography, is you could sit there for hours just extracting ions. And what happened in many of in this plasmid fire is there was many different types of halogenated PAH. So this is dichlorophenanthrene, and you can see it and pick it out. And like I said, when there's tens of thousands of compounds, that can take a long time. So in our lab, we have an FTICR that we've used as a very high resolution instrument, and try to look at a different approach. But just to give you an idea, if you have a mass spectrometer, and that's just normal regular old mass spectrometer that the peak width here is 0.3 Daltons. But when you dial up the resolution, there's actually you know, tens or 20, tens or up many tens of peaks underneath that, which make up this one peak. And so there's a real challenge often at, you know, trying to identify what's really there. So this is that plasmid sample. We took 1 microliter of it and put it on a solid probe into the mass spectrometer. There was actually 3,000 peaks from 150 to 500, so a number of different masses, and it's kind of like, you know, well, it's not a needle in the haystack, it's the opposite. We're really good with our target compounds of finding and identifying the needle in the haystack, but, you know, maybe you want to look at what is the haystack made of, or what's the rest of the haystack, or in that haystack. So one way to do it is using a mass defect plot. And these were originated by Kendrick a number of years ago. And what he did is he took a methylene group, and it works in the usually in the petroleum industry to look at homologs or series of compounds. And if you convert the IUPAC mass to the Kendrick mass by using this fraction, then what happens is, if you want to look at something, so here's an example of methyl, ethyl, and propyl naphthalene, 141.9195, and so you can see that when you do this, the decimal points of the compounds are the same. You can plot them, and they come out on lines, which makes life much easier to identify. Obviously, there's a lot of different isotopes from the elements that we have, and so mass defect is the difference from the nominal mass, and you can see that you know, obviously as you go down here from chlorine to bromine, it increases. They're additive, so if you have 5 chlorines, it's basically 5 chlorine 35s. It'll take this much off from nominal mass, and when you plot this, they all sit on top of each other, making it a lot easier to identify these compounds. So again, this just gives you, this is a simplified schematic of a Kendrick plot, and what happens here is, if you go in this direction, you're adding CH2, and this is the classical Kendrick plot. When you're going down, you're taking away H2, hydrogens. So I'll show some examples in a second to give you a much better idea of what's going on. So here is a classical Kendrick plot, or mass defect plot, to the CH2 unit. So black dots are carbon. If there is a blue dot, it has a nitrogen, red has an oxygen, green O2, and yellow N2. So this is a plasmid sample that had thousands of compounds. You go this way, you're adding a CH2 unit. You go this way, you're taking off an H2 unit. So you can see that this is extremely complex with thousands of compounds. And so when we started to look at it, this is just a small fraction. In that sample, about over 16% of it was pyrenes, and then there was benzopyrrolenes. There was a number of halogenated compounds, and I'll show you that in a minute. And one other way you can tweak it even more and is to, instead of using CH2, Vince Taguchi, one of the scientists in our group, did it for chlorine. So you use the mass of chlorine over 35, and then what it does is it'll align anything that has a chlorine, or you could use anything as long as you could use a bromine or whatever, and it'll rotate this figure so that the compounds you're looking for are sitting horizontal. So again, this, I showed you this top part of the diagram before, and this is almost at 90 degrees. These are all the chlorinated or halogenated compounds. So when we do it to look at the chlorine, so this is a mass defect plot that's been normalized for chlorine. You see all these bands, everything that's horizontal here has chlorines or bromines in it. The bromines kind of sort of sit on the same line. And just to give you an idea, here's dioxin. So you have the the green is the dioxins, and the purple is the substitution for bromine. So here are the thiophene and the, sorry, this is the furans. The dioxins are up here. So the blue is the dioxins, and the red is the bromine substitution. So just to give you an idea, you know, there's about 5,000 bromochloro-dioxins. So all these, what are they, about 30 dots or 40 dots are representative of about 5,000 compounds. And then, you know, here's the anthracenes, same sort of thing. And you can just keep, now we took a spot down that diagram, and as we're moving down now. So, you know, you get the PAH. Dave talked about a whole bunch of PAH, and it's the same sort of thing. There's many different compounds here, and just by looking at it, you can get an idea. You know, you have CL, you know, the non-chlorinated, and then there's 1, 2, 3 chlorines, and then there's substitution from bromines, and for a variety of different compounds. And you can just go on and up down the diagrams, and this just gives you an example. Some of the bigger ones didn't have as much chlorinated substitution on it, but you can see that this was taken from 100,000 resolution. So there basically is, this is pretty much confirmed that this is this compound, unless, I guess this is choranine. So this is choranine unless there's some other organization of the phenyl rings. This is Carl's work, and Carl kind of just took the simplest scenario. So now we wanted to do some confirmation. We had an older time of flight that we converted to a 2D instrument. This runs at about 7,000 resolution. We're trying to get an instrument that has as high a resolution as possible, and someday hopefully we'll get a 2D instrument that can run in 2D time frame at 10,000 resolution or more. To do most of these, as long as you got 10,000 resolution, you can get started. So this is a total ion chromatogram, again, of the plastimate, and then this is just the total ion chromatogram or extracted iongram of the bromo, chlorine, phenanthrene, or anthracenes. We can't tell if it's phenanthrene or anthracene because they have the same molecular weight, but you can see that there's 100 or more of these compounds just by adding chlorines up here and then bromochloro. So we're starting to get some separation. What we did is take a look at, or we actually exposed a number of biota to that ash. And what happens then is these are just hydrocarbons. These are halogenated compounds, and the red is the halogenated compounds that were bioaccumulated or that were taken up by the biota. So you can see there's only a small number that actually bioaccumulates, and that's consistent with things like dioxins or what we found other halogenated PAH, that only a small subset of these compounds bioaccumulate. And so with the flies and minnows, some of them, this was 2378 TCDD, but we saw compounds like tetrachloroanthracene, tetrachloropyrene, trichlorobromopyrene, and you can see you're getting, this is a biosediment accumulation factor. We also did is these are some of the mayflies. Again, these are the chlorinated phenanthrene or anthracene. I think there's 38 peaks, Carl had told me, and there's about 8 or 10 here that bioaccumulate. So this gives you an idea of, you know, the amount of compounds that are present and the ability to look at compounds that may or may not bioaccumulate. So what we did then is start to look for compounds using these mass defect plots in Lake Ontario lake trout, and you can see there's a ton of peaks again, and they were just what you do is you add up the entire mass spectrum, and here it is, about 1,000 peaks with a signal noise of 3 to 1. You convert it and turn it into your mass defect plot. So here it is, the classical Kendrick plot. These are hydrocarbons, halogenated compounds, and there were some siloxanes that had gotten in there, probably contamination. This is a little cut over here that shows that kind of expanded out, and then this is a little bigger. So in Lake Ontario fish, we saw chlorinated terphenyls, DDT degradation products. We're assuming these are anthracenes because we think they would bioaccumulate more than the phenanthrene, but we're not sure. Diphenyl ethers, PCBs, PCNs, some brominated compounds that turn out to be related to some of the Dechlorane compounds that we saw earlier, because here are the fragmentation products, the retrodiels alder fragmentation products. And so when we pull a mass spectrum out, this is 604. This is Dechlorane 604, but we also see a product that's dibromochloro. So the 604 has 4 bromines, and here's the mass spectrum. So in lake trout, we see a tribromo, and this, and we also see a dibromo. So these compounds are present in these samples in the lake trout. We also see tetrachloroanthocene and phenanthrene, and these, I guess, are the exact mass, and you can see the number of isomers that's detected. So just to summarize, the using mass defect plots or mass deficiency is an intrinsic property of polyhalogenated compounds. High mass resolution routinely exploits this property in analysis of polyhalogenated compounds like dioxins, PCBs, or PCNs. We can use the mass deficiency to identify target or non-target compounds. Mass defect plots are efficient tools for identifying unknowns, including halogenated PAH or mixed halogenated PAH, as well as other compounds like these Dechlorane flame retardants. And do you need to use an FTICR? No, you can use pretty much anything that gives you 10,000 resolution or more. It would be any TOF or instrument like an Orbitrap. The high resolution mass spectrometry, I just said that, can be used to identify unknowns. Ideally, you want to have good chromatography to separate out these compounds and identify them. We're able to identify a series of Dechlorane compounds and their degradation products in fish, including Dechlorane Plus 602, 3, and 4 were detected around the Great Lakes, as well as their degradation products. And Dechlorane 602 and its declaration products undergo long-range transport and are present in most biological tissues that we've analyzed. Thank you.